Hi, this is Daryl York. I'm here to talk about HIV and its effect on cell-to-cell communication. HIV, or human immune deficiency virus, slowly destroys the immune system by attacking and killing CD4 cells, or T cells, which are white blood cells in the body that play a major role in protecting the body from infection. HIV uses the machinery of the CD4 cells to multiply and spread throughout the body itself. The 7-step process is called the HIV Life Cycle. HIV may lead to AIDS or acquired immune deficiency syndrome if not treated. With a lower CD4 cell count, one with HIV is prone to many other infections and or cancers. The end result of HIV is a weak immune system and is considered AIDS, which is signaled when one becomes sick to a disease or infection that is usually harmless. This is an HIV cell. It has 5 key components. HIV glycoproteins are embedded into the HIV envelope, which is the outer surface of HIV that houses the rest of its organelles. Inside the envelope rests the HIV capsid, which is HIV's core that holds HIV RNA and HIV enzymes. Within the capsid, there are HIV enzymes, which are those proteins that carry out the steps of the HIV life cycle. And there is also HIV RNA, which is the genetic material being copied. During step 1 of the HIV life cycle, the HIV cell's glycoproteins attach to the CD4 receptors outside of the cell. Next, after HIV attaches itself to a host CD4 cell, the HIV viral envelope fuses with the CD4 cell membrane. This fusion allows HIV to enter the CD4 cell. Once inside the cell, the virus releases HIV RNA and HIV enzymes. After this, the HIV cell uses the enzyme reverse transcriptase to turn HIV RNA into HIV DNA, which enters the cell's nucleus and eventually combines with the CD4 DNA. Once inside the CD4 cell nucleus, HIV releases the enzyme integrase, which HIV uses to insert or integrate its viral DNA into the DNA of the host cell. After HIV has integrated into the CD4 cell's DNA, the virus begins to use the machinery and organelles of the CD4 cell to create HIV protein chains that are building blocks for more HIV. During assembly, those new HIV RNA and HIV proteins move to the surface of the cell through exocytosis and assemble within a vesicle into immature or non-infectious HIV. Finally, the immature HIV pushes itself out of the host cell. Non-infectious HIV cannot infect other CD4 cells, so once outside the old cell, this new HIV releases protease, an enzyme that breaks up the protein chains that combine to form mature infectious HIV. After leaving the CD4 cell, the combined and corrupted DNA is left behind and induces apoptosis, or cell death. Since this is potentially happening to hundreds of cells at the same time, one's immune system could be whittled down to being useless within just a few years. No effective cure exists for HIV, but with medical care, it can be controlled. The medicine used to treat HIV is called antiretroviral therapy, or ART. If taken properly, the amount of HIV in one's blood can become undetectable. If it stays undetectable, they can live long, healthy lives with no real risk of transmitting HIV through sex. Normally, instead of HIV corrupting and destroying the CD4 cell, CD4 are subsets of the human white blood cells that protect the body as the immune system. Clearly, the corruption of these important cells in the body would be very dangerous and is the reason that HIV is so dangerous.